Morphology of fungi. Fungi are eukaryote. They have true nuclei surrounded by nuclear membrane and have rigid cell wall like plant. They are larger than prokaryote with typically filamentous growth. The majority are aerobes. They require oxygen at some stage of their growth. Fungi live everywhere and have very diverse habitat. Most fungi are saprophytes and found on vegetation, decomposing organic matter, soil, and water. Some are parasitic on plants, animals, and insects. Many fungi can produce mycotoxin in food and in animal feed, which can cause toxicosis. Some used in food industries. Saccharomyces used in alcohol, drink, yogurt, bread. Penicillium roqueforti, which is used in producing cheese. Many used in vitamin production. Penicillin chrysogenum used in producing penicillin G. Some are obligate aerobes, other facultative aerobes, and they can ferment sugar and grow anaerobically. Fungi are organotrophs, require organic source of carbon. Fungi are a slow grower in comparison to many bacteria and resistant to antibiotics. Morphology of fungi. Fungi grow in two morphological forms, yeast or molds. Yeast grow mainly by budding, producing spherical to ellipsoid cells 3 to 15 micron in diameter, either single or attached to each other, forming pseudohyphae. They never produce aerial hyphae. This is reflect on morphology of the colony on sugar media, which resembles bacterial growth. The colony are pasty, opaque, and may be white, creamy, yellowish, red, or even black in color. The yeast color generally attain a diameter of 0.5 to 3 mm within 3 days. The mold grows through the development of tube-like extension, forming what is known as hypha. The mold grows through the development of tube-like extension, forming what is known as hypha. The mass of intertwined hyphae is called mycelium. The hypha may be divided into individual cells by septa, which is cross-walled, that present one or two pores. The hypha may present no septa and remain as continuous tube. A portion of the mycelium grow into the medium called vegetative mycelium. Visible part grow above the medium called aerial mycelium. The aerial mycelium gives the colony fluffy appearance and bear the reproductive structure which gives the colony its color. The mold colony varies in its appearance size from few millimeters to few centimeters, so the single colony may cover all the surface of the plate. It may be glabrous, varicose, fluffy. Surface texture may be velvety, cottony, powdery, may assume any color or grade of color known in nature. Some molds have the character of dimorphism, dimorphic molds, as they can grow also in a yeast form. It takes place in tissues. When the mold invades tissue, it changes to yeast form, or in lab when the colony plated on enriched medium, such as brain, heart infusion agar, or incubated at 37 centigrade. The rigid cell wall of fungi consists of chitinous microfibrils embedded in a matrix of small polysaccharide protein lipids. The major polysaccharide cell wall matrix consists of glycan, mannans, chitosan, and galactan. Yeast have soluble peptidomannan as component of their outer cell wall. The outer cell wall of dermatophyte contain glycopeptide that may evoke both immediate and delayed hypersensitivity. The plasma membrane is similar to the mammalian plasma membrane, therefore it has the non-polar ergosterol instead of cholesterol as a principal sterol. This is the site of action of antifungal. Fungal nuclei are variable in size and shape and members. The DNA and the associated protein occur as long filaments of chromatin which condense during nuclear division. The number of chromosomes varies with particular fungus. The fungal cell may contain up to 100 mitochondria.